Hello, good morning, all of you. In our previous lecture, we discussed regarding the fermentation process, which is an ATP generating metabolic process where the electrons are transferred from one compound to other and reducing power in the form of NADPH or NADH is generated. That is later on converted into ATP by electron transport chain. So in metabolism topic we studied different metabolic pathways occurring in different organisms and which lead to synthesis of ATP as well as other intermediate compounds, and these compounds are utilized for biosynthetic purpose for anabolic reactions to synthesize different macromolecules. So we discuss TCA cycle, we discuss glycolytic pathway as well as HMP pathway, ED pathway. So these pathways we studied, and the significance of all these pathway is to generate energy in the form of ATP. Simultaneously, we also studied that whatever intermediate compounds are produced during the biosynthetic or biochemical reaction, these compounds are not wasted. These are utilized by one or other way for the anabolic reaction or to synthesize different cellular constituent or different molecules, macromolecules. For example, to synthesize DNA, RNA, amino acid, etc. So it means that though the biochemical pathways are different in different microorganisms, ultimately the significance is same, that is to generate energy as well as to synthesize precursor compound for the synthesis of different macromolecules. So the significance is almost similar. Now we are going to study another energy generating metabolic process that is called as fermentation. So what is fermentation? During fermentation, it is observed that the electrons are transferred alone with protons from reduced compound or reduced coenzyme, for example NADH or other compounds, to pyruvic acid, and the final electron acceptor get reduced to form various end product. We know in fermentation, you might have discussed in industrial microbiology, the fermentation reactions are those in which energy is generated where the both compounds are organic, means electron acceptor, electron donor in nature, and there is energy generation. So in fermentation process, there is a sometime acid production is there, gas production is there, that is the industrial fermentation. Now here we are going to study how the electrons are transferred and how the energy is generated in fermentation reaction. The important function of fermentation is to regenerate NAD. NAD means nicotinamide adenine dinucleotide, or it is a coenzyme. We know coenzymes and cofactors. Coenzymes are different, cofactors are different. For specific enzyme, specific coenzymes are required or specific cofactors are required. So NAD is the coenzyme for dehydrogenase enzyme, which catalyze dehydrogenation type of reaction. So here the fermentation function is to regenerate or to reproduce NAD, which is required at 6th reaction of glycolysis. We know glycolytic pathway or EMP pathway. In glycolytic pathway, what we studied, glucose, which is a carbohydrate, that glucose is completely broken to pyruvic acid by the set of or by the series of 9 reactions. And pyruvic acid, whenever it is produced, that may be fermented to lactic acid later on in fermentation reaction. So whatever pyruvic acid is produced, that may undergo the reaction by different type of the enzyme, for example lactate dehydrogenase, where pyruvic acid undergo reduction reaction to lactic acid. So this is one of the best example of fermentation of lactic acid that we are going to study further. So in glycolytic pathway, there is a production of pyruvic acid from glucose and there is a continuation of the reaction simultaneously producing ATP or energy in the form of ATP. So microbial fermentation. Microbial fermentation means what? The fermentation is carried out by different microbial species. We know different fermentation products are there. In industry, on a larger scale, different industrial products are produced. It may be ethanol, it may be butanol, it may be beer or it may be wine or it may be yogurt as well as curd. So different types of the fermentation or different pharmaceutical products are also there. So different fermentation products are produced in industry. So these fermentation reactions are conducted by different types of microorganisms. In fermentation media, we are going to add inoculum or culture, microbial culture. And this microbial culture, they are conducting fermentation process to produce a typical end product, that is the fermentable product. So the fermentation process is used to produce either dairy products, soya sauce, different types of the pickles, ethanol, butanol, as well as organic acids and different pharmaceutical products. Pharmaceutical products means antibiotics and other drugs. So it is also useful for diagnostic purpose. So diagnostic kits are also there and we can use different microorganisms for this purpose. So entirely we are going to employ different microbial species to carry out fermentation process. Means what? Whenever we want to produce organic acid on a larger scale, then we can employ some kind of bacteria in fermentation process. Whenever we want to produce organic acid, for example lactic acid, then we can use certain species of lactobacilli or other bacteria which can convert pyruvic acid to lactic acid. So it means lactic acid fermentation is carried out by bacteria. Similarly, in case of production of pickles as well as soya sauce and other pharmaceutical products, we can use different types of microorganisms which are producing specific enzymes to convert the raw material into product, that is the fermentable product. So in this way we can utilize microbial species for the fermentation process. So in this slide different fermented products are shown here. It includes yogurt or curd. It includes different types of the drinks or beverages are also there. Pickle is also there. So these are the examples of fermented products. You might be knowing idli, dokla, jalebi. These are also fermentable products. Curd is also best example of fermentable products. So idli batter, curd as well as cheese, beer and pickles, soya sauce, these are all fermentable foods. What are different types of fermentations? The next point we are going to study types of fermentation. There are different types of fermentation. For example, lactic acid fermentation, butanol fermentation, ethanol fermentation, etc. The most common and old type of fermentation is lactic acid fermentation. See here, the lactic acid is organic acid. So lactic acid is produced by fermentation reaction or chemical reaction where we can use pyruvic acid and we can convert pyruvic acid to lactic acid with the help of bacteria. So lactobacillus species, Bifidobacterium species, Leuconostoc species, these are some kind of bacterial species which can be used for the fermentation reaction to produce our interested fermented product. It may be lactic acid, it may be any kind butanol, it may be ethanol, it may be organic acid, or it may be pharmaceutical products. Here I have taken the example of lactic acid. So lactic acid is also produced from pyruvic acid by the reduction reaction. What is the reduction reaction? Addition of hydrogen. So here pyruvic acid is converted or reduced to lactic acid. The enzyme will be lactate dehydrogenase. Lactate dehydrogenase remove 2 hydrogen from reduced NAD means NADH. NADH हा reduced compound आहे. NADH plus H plus मधले 2 हायड्रोजन रिमूव्ह होतात आणि ते 2 हायड्रोजन पायरुविक ॲसिडमध्ये ॲड होतात. पायरुविक ॲसिडचा फॉर्म्युला काय आहे? CH3 सिल्वर बॉन्ड O COH. पायरुविक ॲसिडमध्ये 2 हायड्रोजन त्या ठिकाणी ॲड होतात. सो व्हॉट इज द फॉर्म्युला हियर? CH3 CHOH COH, that is lactic acid. So in this way pyruvic acid will be converted to lactic acid. The enzyme catalyzing this reaction will be lactate dehydrogenase. लॅक्टिक ॲसिडमधले 2 हायड्रोजन ते NAD कडे जातात. NAD रिड्यूस होतो, NADH तयार होतो. त्या NADH मधले हायड्रोजन हे तिथे पायरुविक ॲसिडमध्ये ॲड होणार आहेत आणि पायरुविक ॲसिडचं कन्व्हर्जन लॅक्टिक ॲसिडमध्ये होतं. So this is the reaction. Lactic acid is used. What is the application of lactic acid? Now we produce lactic acid, no doubt, by fermentation reaction. But what is the use of lactic acid? Lactic acid is used to flavor and preserve dairy and vegetables, and ethanol fermentation is used in bread and beer production. So lactic acid, whatever it is produced by fermentation reaction, this lactic acid will be used further either to preserve certain dairy products or to preserve vegetables. Similarly, ethanol fermentation is also one of the type of fermentation where we produce ethanol by fermentation reaction, which can be used for beer production and in the bread production also. Then now we are studying here lactic acid fermentation, homolactic acid and heterolactic acid fermentation. So lactic acid fermentation, it is carried out by some bacteria, for example bifidobacteria and lactobacillus species. 2 bacterial genera are important. One is bifidobacterium and another one is lactobacillus species. We know lactobacillus are present abundantly in curd sample. Lactobacilli convert sugar to lactic acid, so the curd is sour in taste. That lactobacilli are used for lactic acid fermentation process. So there are 2 different types of microbial lactic acid fermentation process. 2 पद्धतीनं 2 फर्मेंटेशनच्या प्रोसेसमध्ये लॅक्टिक ॲसिड तयार होतं. एक म्हणजे होमोलॅक्टिक ॲसिड फर्मेंटेशन, सेकंडली हेट्रोलॅक्टिक ॲसिड फर्मेंटेशन. So by any fermentation method, there is a production of lactic acid. Either there will be homolactic acid type or there may be heterolactic acid. मग या दोन्हीमध्ये काय डिफरन्स आहे? इन homolactic acid fermentation, मेजर फर्मेंटेबल प्रोडक्ट इज लॅक्टिक ॲसिड. होमोलॅक्टिक ॲसिड प्रोसेसमध्ये काय होतं? ज्या काही बॅक्टेरिया त्यामध्ये भाग घेतात त्या बॅक्टेरिया काय करतात? लॅक्टिक ॲसिड हा मुख्य प्रोडक्ट तयार करतात. हेट्रोलॅक्टिक ॲसिड फर्मेंटेशनमध्ये ज्या बॅक्टेरिया इन्व्हॉल्व होतात त्या बॅक्टेरिया काय करतात? लॅक्टिक ॲसिड तयार करतात, पण त्याबरोबर इथेनॉल, कार्बन डायऑक्साईड हे पण प्रोडक्ट्स इक्विमोलर कॉन्सन्ट्रेशनमध्ये तयार करतात. इट मीन्स देर इज अ स्मॉल डिफरन्स इन होमो अँड हेट्रो फर्मेंटेशन. इन homolactic acid fermentation, lactic acid will be the major product of fermentation, whereas in heterofermentation, along with lactic acid, carbon dioxide will be also there, ethanol is also there, and both, that means ethanol, CO2 and lactic acid, these are products in equimolar concentration. That is the heterolactic acid fermentation. So in homolactic we will discuss first. Homolactic acid fermentation, in this type of fermentation, all glucose, starting compound is always glucose. So glucose is converted to lactic acid. All glucose molecules are converted to lactic acid. So this fermentation is carried out by bacteria like Enterococcus, Lactobacillus, Streptococcus, Pediococcus. So Lactobacillus is also there, Enterococcus is also there, Streptococcus is also there, Pediococcus is also there. So these species, these bacterial species, they are conducting homolactic acid fermentation. Enterococcus, Streptococcus, Lactobacilli and Pediococcus. The two pyruvic acid molecules formed at the end of glycolytic pathway, means at the end of EMP pathway, pyruvic acid molecules are produced. We know very well. So these pyruvic acid molecules are converted or reduced to lactic acid by the enzyme lactate dehydrogenase. Lactate dehydrogenase is the enzyme. So this enzyme catalyze conversion of pyruvic acid to lactic acid, where two hydrogens are removed and these hydrogens are added or accepted by pyruvic acid. So pyruvic acid get reduced to lactic acid. The reaction is shown in this slide. Pyruvic acid formula is shown here, CH3, C double bond O, COH. This is pyruvic acid. Pyruvic acid is reduced by, reduce means addition of hydrogen. दोन हायड्रोजन कोण देणार आहे? NADH प्लस H प्लस. NADH प्लस H प्लस मधले दोन हायड्रोजन जाणार आहेत. म्हणजे काय उरणार? फक्त NAD. That is shown here. So two hydrogens are removed by the enzyme lactate dehydrogenase, that is shown here below the arrow. So this enzyme removes two hydrogen from reduced form of NAD. म्हणजे NADH प्लस H प्लस मधले दोन हायड्रोजन जाणार आहेत आणि काय उरणार? फक्त NAD. ते जे दोन हायड्रोजन्स आहेत ते कोण ॲक्सेप्ट करणार? pyruvic acid. So two hydrogens are added in pyruvic acid molecule. मग pyruvic acid मध्ये दोन हायड्रोजन ॲड केल्यानंतर फॉर्म्युला काय होणार? CH3 CHOH COH. दोन हायड्रोजन फक्त ॲड करायचे. CH3 CHOH. डबल बॉन्ड ज्या ठिकाणी आहे त्या ठिकाणी दोन हायड्रोजन ॲड झाले. म्हणजे फॉर्म्युला काय झाला? CH3 आहे तसंच CHOH COH. That is lactic acid. So in this way the enzyme lactate dehydrogenase catalyze the transfer of hydrogen atoms from NADH plus H plus to pyruvic acid. pyruvic acid हा हायड्रोजन घेणारा आहे आणि NADH हा हायड्रोजन देणार आहे. So whenever pyruvic acid accept hydrogen, the pyruvic acid will be converted to lactic acid. So it will get reduced to lactic acid. Pyruvic acid get reduced to lactic acid. रिडक्शन म्हणजे ॲडिशन ऑफ हायड्रोजन आणि ऑक्सिडेशन म्हणजे रिमूव्हल ऑफ हायड्रोजन. ऑक्सिडेशन मीन्स रिमूव्हल ऑफ हायड्रोजन. रिडक्शन मीन्स ॲडिशन ऑफ हायड्रोजन. So in homolactic fermentation, 2 ATP molecules are produced per glucose molecule, which is converted to pyruvic acid. एक ग्लुकोज मॉलिक्यूल जेव्हा पायरुविक ॲसिडमध्ये कन्वर्ट होतो तेव्हा 2 ATP मॉलिक्यूल पर ग्लुकोज मॉलिक्यूल तयार होतात इन homolactic acid fermentation. So in this way we discuss